Okay, shall we get going? So I think we've just had the last few talks have really led up to what I hope we want to discuss in this session, which is about novel therapies, but also something about current therapies and the process we all have to go through in making decisions. In many ways, it's good to have choices, but choices mean we have to make decisions, and that's not always easy. And you've just heard just now that we need information, and we need information about what's coming down the track, not just what's available now. And this applies to not just to clinical trials, this applies to everyday therapy. And the biggest decision-making that most of us in this room have had to do, not including myself, I'm talking about the families, is about transplantation, for instance. And you've just heard from Henkian what the data are. And at least we can make, in the context of transplantation, we can make decisions on good quality data now because we've got large numbers of patients and we can see exactly what the risks and benefits are. And it's clear that transplantation has got many benefits, but it's actually got considerable risks as well. And hidden within those data is the simple fact that if you're going to sign a consent form for your child to go to transplant, you're signing a consent form that has at least a 10% risk that the child is not going to leave hospital. And that's a really horrible, horrible thing for anyone to do. And, I have every sympathy for people who've had to face that. I hope that everyone's felt that they're doing that with the agreement of the people that are looking after them, and they're not being forced into that decision, or they're not being left to make that decision on their own. And that's the way we would aim to have consent, is because everyone agrees and everyone understands, and no one feels that they've been left to make a decision on their own or had a decision forced upon them. But it is a horrible situation to be in. And of course, liver transplantation has a degree of irreversibility about it, so that makes it a lot more difficult. In the last few years, as you know, we've done these clinical trials with IBAT inhibitors, and that's actually, we've been very lucky because those drugs actually had a lot of clinical data before we gave them to any children with liver disease. They've been used extensively in adults in clinical trials, so we had lots of safety data. And we were all reassured by the fact that the drugs are minimally absorbed. They're basically only acting in the gut. They're not having much systemic exposure. They may have systemic effects, but the drug themselves is only working in the gut. So most people who were involved in the clinical trials and decision-making there didn't face the complexity of decision, I don't think, in most cases, that we're having to make in some of these new therapies that we've just been talking about, where we're going to have to make more difficult decisions. So if we take a step back about how we're going to get any of these therapies into clinical practice, we have to get as much preclinical data, so that's using in vitro in the lab, and then in experimental animals, and most often, I hope it's obvious that has to usually go through some small mammalian system, whether it's mice or larger small mammals, and very often it has to go through non-human primates in small numbers, but I think they are much closer to us, and that is feels horrible that we have to use non-human primates for studies still, but very often if we've got a completely novel therapy, we have to do that before we can give it for the first human experiment. No one wants to be an experiment, but without experiments, we will not progress medicine. The first liver transplant was an experiment, and it was only by somebody being prepared to do that we are where we're now with liver transplantation. So I think we're making decisions all the time, and it's through clinical trials that we will move forward. And the difficult thing about clinical trials is that we are, and it just came to the end of the last discussion, is we have unknown risks, and that's the big difference between decision-making about transplantation, where we've got really good data, we know what the risks are, we know what the benefits are. And even if the risks are considerable, we can see the benefits are enough and we can make a decision. But by definition, the whole point of clinical trials is we don't know all the risks and we don't know all the benefits. And so that puts us in a much more difficult position, us as clinicians, you as families, when we're making decisions about entering clinical trials. So there's lots of people involved in clinical trials. There's the families, there's the clinicians, there's the scientists who developed the technology, there's somebody who's paying for it, usually through a pharmaceutical company, maybe through an academic institution. And at the end of the day, of course, the object of the exercise is to get a new treatment. And as you know, in every country, the treatments are regulated by appropriate authorities. In this country, it's the FDA. And the FDA are there for a very important reason. The first reason they're there is patient safety, and to make sure that we're not doing stupid things, and to make sure that any experiment we do has a realistic chance of getting useful data, which will then empower us to make decisions about whether a therapy is an appropriate therapy to use in the future. And then so they have to approve the experiment and then decide whether the evidence at the end of that trial is enough to move forward and use something therapeutically in the future and license it or not. So there's many, many stakeholders in moving this forward. And it's possible, and I think it's definitely true, that the families haven't always been involved in that process as much as they should have been. And that's not just because we should do it. I think if we involve the families right from the beginning, we can probably do it better. So that's really one of our main objectives today, is to start at least thinking about this whole process from a family point of view, not to make lots of decisions today, but to at least start the thinking about the concepts that are involved. And then that will help us do a number of things. I think we can have some collective decisions as a community about how we'd like to see some aspects of trial design, and we can go into the different parts of what makes a trial, but also have a body of people who are available, so when a company, for instance, wants to design a study, they can come to the network and say, right, we want to work on a trial for this condition with this sort of therapy, and there's people available that they can talk to and help them come up with a study which we're all happy with. That's the best study we can think of, given all the information that's available. And then also then go forward and help get the money for that and get the approvals for that study. And as I've already mentioned, the FDA is an important partner in all of this because they have a very important function in this. And we were hoping that Dr. Ruby Mehta would be available today, but unfortunately she's otherwise engaged, but she's really sorry that she's not here. And we have, Emily and I have talked to her beforehand. She will be on the chat, and I've got a few things of which we got from our discussion with her, which I'll go through a little bit later. But Ruby is a pediatric gastroenterologist and hepatologist who works full-time for the FDA these days and is one of many people who are very important in getting clinical trials approved and getting ultimately drugs approved after that. But she is at least she has come from the same background as many of us and is involved in that process and has been extremely helpful. So it's a great shame she's not here today. But as I say, she's been involved in this process and we've had previous conversations, but I will go through some of the things she wanted to talk about here a little bit later this afternoon. So we talked a little bit about the novel therapies. Zaki was talking about that. We talked about transplantation, and we want to get involved in the discussion about the thoughts in the course of this afternoon. What we're going to do first, though, is we're going to have many of you will know Lorenzo D'Antiga, who is a colleague of ours. He's a pediatric hepatologist who runs a center in Bergamo in Italy. So he's a PFIC expert as well, but he has recently been involved in a clinical trial for Crigler-Najjar syndrome, which was mentioned earlier on today. It is a liver disease. It causes unconjugated jaundice. It's not really cholestasis, but the point is that he has got first-hand experience of going through the consent process and explaining all the things we've just been talking about with families who have ultimately agreed to go into a clinical trial using, as you'll see here, an AAV therapy, which we've just talked about earlier on. And I thought it would be a good starting point if we can kick off with Lorenzo's thoughts and comments, having recently gone through that process. Lorenzo, can you hear us? I can. I'm getting nods from most places in the room. Does that sound like a plan, Lorenzo? Can you take it from there? I think you're a bit quiet, and Lorenzo, just for 1 second, you're a bit quiet. Can we get the volume up at all? I'm standing next to a speaker, so I think I can hear you better. That's fantastic. Lorenzo, halfway in between would be even better. That's too much. Just go a bit further away. Keep going. That's great. Thank you.
Inside the capsid. What we do is a single peripheral vein infusion, and the vectors get into the liver because this capsid has a special tropism to the liver cells. And when the vectors are in the liver, in the sinusoids, they enter the hepatocytes, they infect the hepatocytes. After that, the vector that you can see here as a capsid and a transgene inside in the endosome is released in the cytoplasm. The capsid, the vector, gets into the nucleus. The capsid opens and releases this transgene. The transgene is a fragment of DNA, including the sequence that is defective in the patient. So this fragment then gets this circular shape and it's called episomal. And the good thing in AAV is that this DNA does not integrate into the host genome, meaning that this is a sort of a small chromosome that stays next to the patient's chromosomes without modifying them. But then this DNA is recognized by the cell system. It is transcribed into mRNA that gets to the ribosomes, which is the machine to translate the mRNA into protein. And in our case, the protein produced is UGT1A1, which is a protein missing in Kriganajar. But it might be the same process may also come to synthesize proteins that are meaningful for PFICs. So just a little bit of the results of my experience with gene therapy for Kriganajar. This is a multi-center trial, and what I want to highlight here that this is a no-profit study. The study has been sponsored by Genethon, that is the spin-off of Telethon, so a no-profit organization, and supported by the Horizon 2020 plan. But when we talk about bringing a drug to the market, we have to consider that independent research or no-profit research is almost impossible. This is really exceptional to raise enough money to be able to do a no-profit study like this. So I think we have to realize that bringing drugs to our patients involves liaise with companies and trying to get the best for our patients with a liaison with for-profit companies. Kriganajar syndrome, just to tell you what type of disease this is a condition in which bilirubin cannot be processed to be eliminated by the liver and remains in the bloodstream in the form of unconjugated bilirubin, whereas patients with PFIC have conjugated bilirubin. Phototherapy, these blue lamps, can control partially the levels of unconjugated bilirubin, but these lamps should be applied every night for 8 to 12 hours, the whole night, with the patient naked under the lamp, in winter, in summer. But this phototherapy loses efficacy after puberty. Unconjugated bilirubin crosses the blood-brain barrier, meaning that from the blood it can get into the brain, the central nervous system, and can cause neurological damage. But it is, even if we can control that, these patients have a very poor quality of life because they cannot travel, they have to sleep under the blue lamp every night. The only curative option is liver transplantation, and this has been the treatment offered so far. So I think Richard, and also in the previous talks, it has been shown a little bit of the research process from preclinical phase to phase 1, phase 2, phase 3. I would like to emphasize phase 1 clinical studies. Phase 1 clinical studies are those studies in which a drug that has been studied in cells or in small animals is administered for the first time in humans. So it is the phase in which we introduce a new drug, but we really don't know for sure. We don't know exactly what can be the side effects. Phase 1 studies involve very few participants, usually 10, 15 participants, and is primarily meant to assess safety, toxicity, and tolerability. Nowadays, we tend to combine the two phases, phase 1 and phase 2 studies, with a little bit more patients because we believe assessing safety only is insufficient. We want to see also some efficacy. So usually these studies are phase 1-2 studies with very few patients assessing primarily safety and toxicity, but also partially efficacy. What is important to know is that regulatory agencies always want that these treatments are administered to adults. So the phase 1 studies usually start with older patients, ideally adult patients, because, with the aim of protecting children and young ages. So the trial you will see here, the Krieger-Najjar, involved young adults, and only after that phase, now we can go to pediatric patients. This is the common design of a phase 1-2 clinical trial. The bars are the group of patients, the first cohort, then there is a second cohort, and the third cohort. As you can see here, in the first cohort, there are only 3 patients. These 2 cohorts are meant to test different doses of the drug. They are called escalation trial. So the first group of 3 patients got a lower dose of the vector. The second group of patients got a higher dose. And the third got the selected dose. The idea is to select the dose that is effective without being toxic. So the best balance between side effects and efficacy. What is important to know is that in every trial, but especially in phase 1 clinical trials, after the first 2 patients are treated, there is a group of experts, which is called Data Safety Monitoring Board, DSMB, that very accurately evaluates the side effects, the efficacy part. So even 1 or 2 patients are strictly monitored immediately by a board of experts, and you cannot go ahead with other patients until the DSMB has considered safe the trial. So these are the results of this trial. On this left side, you can see these graphs showing bilirubin decreasing remarkably from the start. It is a very fast decrease in bilirubin, which is sustained up to several weeks. And another good thing is that these patients who almost invariably have abnormal liver function test, ALT, see they have an improvement of ALT that gets down to normal levels, as you can see in these patients. And this is Alessia, who granted me this nice picture. She was very clever. She got a picture of herself before and after the treatment to show how the jaundice was completely cleared, and actually very quickly. So what are the advantages and the disadvantages of AAV gene therapy? The advantages is, if effective, transplantation is not required anymore. The disease can be corrected by a single infusion. This is a one-shot treatment. The virus used circulates in our communities, but it is harmless. And I can tell you that really the experience made in the last 10 years with liver-directed AAV gene therapy has shown that it is really very safe. This type of gene therapy does not modify the patient DNA, so there is no risk to have problems. In general, modifying the DNA may cause what we call insertional mutagenesis. That is something in keeping with the risk of developing cancer. The disadvantages is that only one infusion is possible, although we're trying to develop some strategies to make more infusions. After the first infusion, there is immunity arising, so the second infusion is completely neutralized. In young children, growth can reduce the efficacy, And the presence of advanced liver disease may reduce efficacy as well and increase toxicity. If there is fibrosis or cirrhosis, usually the efficacy is expected to be much less and risk of toxicity higher. The patient should receive immunosuppressive drugs because this treatment triggers some immune reaction. And immunosuppression is usually for 6 to 12 months, but no longer. Very high doses may cause liver toxicity and a condition called thrombotic microangiopathy. So it was mentioned before that AAV is used also for vascular disease. In this or in SMA, for instance, or other muscular diseases, it is used at extremely higher doses than for liver-directed correction. And only in these cases, some major complications have been recorded. So let's focus on phase 1 studies and informed consent. If you read a study protocol, probably you have seen this because you've been enrolled in some trial. You will read that the principal investigator has to obtain a voluntary written consent from each patient, and after an appropriate explanation of aims, methods, risks, the patient will be informed that he is free not to participate in the study and that can withdraw consent at any time. The records may be examined by competent authorities and authorized persons, so the information can be available to third parties, but they will be kept strictly confidential. Then there is a written informed consent following a complete verbal information, and the patients and the parent have to be given ample opportunity to inquire. But let's go to something more practical. And what is my view? What should an investigator consider to assess risks and inform properly the families? I think we should start following the patient before the trial begins, possibly, because you need to connect to the patients, get known to the patients and the families. If possible, we should try to join the advisory board of the research body, a company that is running the study to evaluate all the steps of the process. This is what I try to do. If we know the preclinical results, so the results of toxicity studies, for instance, we will be able to consider the risks, the clinical risks much better. We should try to mitigate the exposure of the patient to risks and, for instance, try to reduce the number of follow-up visits if possible. And explain the process of research in phase 1 studies. These, as we were saying, are studies in which a drug is administered for the first time in humans. We should assess carefully the possible adverse events, their reversibility, explain the trial follow-up schedule. Probably you have seen a follow-up schedule. This is an example with all the tests done over time, different weeks. It might be difficult to understand, but investigators should explain carefully what is the engagement of the patients and the time that is required for the trial. What should a doctor communicate or try to communicate to the patients and the families before enrollment? It is certainly important to join the patient advocacy groups meetings. This is what I've been doing with the Krieger-Najjar community. Every year they have a nice meeting like yours, and I've been attending their meetings every year. Last was 2-3 weeks back to update them and stay together. Explain the criteria to select the first treated patients. You know, very often there are many patients that are eligible, but in a phase 1 study, as I was saying, there are really a handful of patients treated. So you try to select those that are, more likely to have no side effects, better efficacy, in the best position to be treated, to be selected. The first treated patients may not have personal advantage. This is the risk of experiences a new drug, but they open a path to other patients. And this is what one of my patients said. She was the very first one. I accepted to be the first patient, thinking also about the benefits this treatment may offer to other patients. I think generosity and altruism also is involved um, in these type of trials when a patient family decide to take part. What are the advantages of enrollment in phase 1 trials? Well, the drug will come to the market several years later. So very likely a patient that is offered a phase 1 trial will never have the chance to use the drug, especially if the disease is progressing. Being the first patient represents a risk, but often is the only chance to benefit from the new treatment. This is particularly important in diseases that are rapidly progressing and that are very severe. During the trial, there is the need to communicate to the patient. For instance, make sure there is direct and fast communication with the patient family. Try to make the patient feel safe and comfortable, and organize a proper safety net in case any adverse event may arise. For instance, I was in touch with the patient GPs, the patients were taking blood tests, they were looked after, at home during the trial and coming only for the follow-up visits. But we were in touch. I knew how to get in touch with them, and I kept communicating personally with my patient. Actually, we talked on WhatsApp. They had my mobile number, so we were just chatting together. They said I became actually their uncle, because sometimes they would also send some text to say, hello, I'm all right. And they were having blood tests at the local laboratory on Wednesdays and Saturdays, and immediately scanned and sent over on my WhatsApp. So I could decide whether, for instance, to increase immunosuppression or do something different. Follow carefully adverse events and results. For instance, once one of my patients had a terrible headache. She went to the emergency department in her city, and she called me, and I talked actually to the doctors over there just to understand if that was an adverse event and what was going on. What is also important is to interact with the trial promoter to suggest amendments to the protocol. For instance, at the moment, I have 2 trials on hold because I think we need to amend the protocol, because we can increase the benefits and decrease the risks to the patients. And in phase 1 clinical trials, it is very likely that you need to amend the protocol because it is the first time you apply the drug. So although the companies get cross if you stop enrolling patients, but if you believe you should get amendments to the protocol, although they're expensive for the companies, you should stop enrollment and push to amend the protocol. And then at the end, what happens is that with these very special patients in phase 1 clinical trials, you become really a friend. You make friends. During the infusion, I was a little bit afraid of adverse events. So this is the time of infusion. I sat on their bed, and we decided to play cards during those 2-3 hours because I just wanted to be there because I was scared. But I can tell you that nothing happened at all. Nothing happened, like infusing a normal saline. It was nice. I was saying I've become the uncle of these young ladies, and I had the pleasure to get invited to Gaia's wedding, and also Stefania had the chance to stop phenobarbital. She was on phenobarbital, and to get pregnant. She sent me this message a few days back. Uovo di Pasqua con sorpresa means Easter egg with a surprise inside. At Easter we have the tradition. I don't know if you have it, to give chocolate eggs to the kids, and there is a surprise inside. And she was obviously referring to her being pregnant. She's supposed to deliver in a few weeks. But thank you very much.